Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. Today we are going to discuss the histology of small intestine. So a little bit about its introduction. The small intestine is about 6 meters in length, extends from stomach to colon. It is highly coiled, primarily meant for absorption and digestion. It consists of duodenum, jejunum and ileum, and this is the diagrammatic presentation. You can see it extends from this esophagus, from the stomach to the large intestine. This is the duodenum, then jejunum and ileum. So duodenum is 25 centimeter long, jejunum is 2.5 meters long and ileum is 3.5 meters long. Little bit about the mucosa. As all other tubular parts of the GIT, the small intestine also consists of the 4 layers. These are the mucosa, the submucosa, the muscularis and the serosa. So in case of mucosa, there are certain small special features which are present in small intestine. One of these features is are the Plica circularis, also called the valves or the Kirk rings. These valves of Kirk ring are the permanent transverse mucosal folds with the core of the submucosa. So you can see these transverse mucosal folds with the core of the submucosa. These folds are permanent and do not disappear on distension. They cover nearly half to 2/3 circumference of the lumen. This is also important. You can see many of them ending over here. So so these cover the half to 2/3 circumference of the lumen. The small intestine starts 5 to 6 centimeter from the pylorus, most numerous in the distal duodenum, and become reduced in size in the middle of the ileum. The function shows down the passage of the contents and increases the surface area of absorption. So you can see these Plica circularis. These are most numerous in distal part of the duodenum especially, and they gradually decreases in the number till the middle of the ileum, and the function is to slow down the passage of the contents from the duodenum and these other parts of the small intestine. So this is the histological diagram, which is H&E stain section of the small intestine, that is jejunum. In this part you can see this is the Plica circularis. This is the fold of the mucosa with the core of the submucosa, and this mucosa consists of the epithelium, the lamina propria and muscularis. And again this mucosa shows some special features, that is surface show elevation. These are finger shape or leaf-like elevations forming the villi, and the surface cells are covered by the microvilli, and these epithelium dips inside to form the mucosal glands present in the lamina propria. And then is the muscularis mucosae, which you can see over here. This line shows a circularly arranged smooth muscle cells of the muscularis mucosae. This area shows the lymphatic nodule, and this is the lamina propria, and then this submucosa, after which there is the serous. And this is the submucosa, and this is muscularis externa, and after this there is the serosa. So detailed about the mucosa. Special feature of the mucosa are the villi. You can see these are the villi. These are the surface projection. The plica are covered by the villi. These are either finger-like or the leaf-like projection, and their core is formed by the loose connective tissue of the lamina propria, extends from the mucosa to the nearly 0.5 to 1.5 millimeter height, and they also contain blood vessels and lymphatics. The special feature is the lymphatic capillary. This green color show the lymphatic capillary, which is present over here. In addition to this lymphatic capillary, large number of the smooth muscle cells are also present, which are derived from the muscularis mucosae, and the large number of the blood vessels are present. This depression shows openings of the mucosal glands, which are present over here. Here is the muscularis mucosae. Here the mucosa finishes, and then is the submucosa, and next is the muscularis externa. So core of the villus consists of extension of the lamina propria with the network of the fenestrated capillaries and central blind acting lacteal which is present over here. Surface of villi are covered by the epithelium. This epithelium is a simple columnar in nature with goblet cells and large number of the microvilli. The goblet cells are seen between the microvilli. So this is at the higher magnification a section through the villus, and in this section you can see large number of the simple columnar epithelial cells are present. In addition, the goblet cells are present. You can see mucous granules over here, mucinogen granules present over here. The microvilli are present over here. We are forming the brush border, and in the center of the villus you can see lamina propria where a blind acting lacteal is present, and the large number of the cellularity in the lamina propria. These all feature shows the that the surface cells are absorptive in nature. Again, another very beautiful diagram, the H&E stain section. Little bit clearance, you must understand that this is the surface, and this line shows the brush border which is formed by the microvilli. This hole from here up till here, this is the whole microvilli, and this one is the core of the villus, and this central area is the central lacteal vessel, and here you can also see the smooth muscle cells of muscularis mucosa are present. Large number of the nuclei present in the lamina propria as well. These are the columnar cells of the surface epithelium, and these are the goblet cells which are present in the surface epithelium. So you must be clear, this is the central lacteal capillary. Some of the students misunderstand that this may be the apical surface, the luminal surface. This is not the luminal surface. You can very clearly see brush border over here, and this is the luminal surface. This is the cross section taken from the surface of the villi. This is the tip of the villus, another tip of the villus, another tip of the villus, and these villi are covered by the simple columnar epithelium with goblet cells, and core of the villus is formed by the lamina propria. Microvilli, major application of the luminal surface from this striated border. Increase the surface area as much as 600 times. You can see these are this is again same diagram. This is the villus and the simple columnar epithelial cells. This one is the villus, and epithelial cells are lined by the microvilli which are forming this striated border. In between the base of the two villi, the epithelium invaginate in the lamina propria to form the intestinal glands or crypts of the lubricant. So you can see over here these are crypts, and these crypts contain goblet cells, Paneth cells, and enteroendocrine cells. So these are the intestinal glands or crypts of the lubricant. Another beautiful diagram, the intestinal glands are present over here. These are the villi. This villi dips down inside, forming the intestinal gland. This is the core of the villus which is formed over here by the lamina propria. The clear area show the central blind acting lacteal capillary. These are the intestinal glands which are present, and this is the muscularis mucosae. So simple tubular glands extending from the muscularis mucosae to the whole thickness of the lamina propria and open at the base of the gland. You can see they are opening over here. So now we move on to the cells of the mucosa. Nearly 7 types of cells are present in the mucosa. These are enterocytes, which are simple columnar epithelial cells, the goblet cells, which are mucus secreting in nature, Paneth cells containing lysozyme present at the bases of the glands, enteroendocrine cells which secrete different enteroendocrine hormone. The M cells are modified enterocytes which are present over the surface where the lymphatic nodules are present, and these M cells are acting as the antigen presenting cells. Then the stem cells which are forming all these types of the cells, and sometimes the occasional intraepithelial lymphocytes are present as well. So this is the diagram of the enterocyte before process of absorption and after the process of absorption. So in this diagram you first see this at the surface. You can see large number of microvilli which are present over here, the tight junctional complexes which separates the liminal compartment from the lateral compartment, the lateral plication of the surface, the basally located elongated nucleus, the Golgi apparatus, mitochondria, smooth endoplasmic reticulum, also the rough endoplasmic reticulum. In this diagram you can see large number of the secretory absorptive vesicles which are present over here. You can also see these absorptive vesicles are released into the intercellular lateral compartment and also into the basal compartment, and they enter either the blood vessels or the lymphatic capillaries to be transported into the general circulation. Another beautiful diagram through the goblet cells. This is a section of the goblet cells having blunt microvilli over here and large number of the mucinogene granules, basally located nucleus, also the mitochondria, smooth endoplasmic reticulum and rough endoplasmic reticulum with Golgi apparatus. This is the section through bases of the intestinal glands or the crypts of the lubricant. In this you can very clearly see these cells. These are the Paneth cells. These Paneth cells contain lysozyme containing granules which stain which show dark eosinophilia in H and E stain, and you can easily identify these Paneth cells in the bases of the glands as well. The same diagram which I previously shown you, this is the basis of the gland, and here you can see the Paneth cells. Another very beautiful diagram showing the surface epithelial cells. Also you can very clearly see the microvilli which are present over here, and this line show the basal bodies which are forming the microvilli. Bases of the gland showing the Paneth cells in addition to the goblet cells and the enterocytes. Another diagram showing the goblet cells, the enterocytes and the Paneth cells present in the basis of the intestinal glands or crypts of lubricant, and this one is the lamina propria. Another diagram to the villus of the small intestine. So simple columnar epithelial cells called enterocytes. This is occasional intraepithelial lymphocyte. These are the goblet cells which are present over here, and this is the lamina propria. This is the central lacteal capillary over here. Again cross section through base of the gland showing lysozyme containing Paneth cells in it. So now we do different features of all parts of the small intestine one by one. First of all, we will look into the duodenum. This is a C shape and about 12 centimeter in length. It continues from the pylorus of the stomach and continues as the jejunum. It consists of the foliar, the mucosa, submucosa, muscularis externa and serosa as in the other parts of the GIT. So this is the diagram through the surface. These are the villi of the duodenum formed by the epithelium with the simple columnar with the goblet cells and microvilli. These are intestinal glands of the lubricant which are present. Then the muscularis mucosae and lamina propria with occasional submucosal Brunner gland and the muscularis mucosae. Now the very beautiful diagram showing all the parts of the duodenum. Here you can see the mucosa. Here the mucosa, this green dotted line show that the mucosa finish over here, and this is submucosa which shows a large number of the Brunner glands. These are branched tubulo-alveolar type of the gland which are mucus secreting in nature, and this is the special feature of the duodenum. And the presence of the Brunner's gland, you can easily identify that this is the duodenal part of the small intestine. So function of this gland is to secrete the alkaline mucus to neutralize the acidic secretion of stomach, and it secretes human epidermal growth factor as well. Duodenum, then the muscularis externa. This muscularis externa again consists of inner circular layer and outer longitudinally arranged smooth muscle layer, and next to this is the serosa or adventitia, as most of the duodenum is retroperitoneal, so it is lined by adventitia as well. Some parts which has peritoneal covering are covered by the serosa. So the duodenum in summary, mucosa, which is having the villi, crypts of lubricant, goblet cells, Pennant cells, and microvilli. Some mucosa with the presence of the Brunner gland, muscularis mucosa similar to other parts, and serosa similar to other parts. Next is jejunum. Same feature as the duodenum, but the difference is that it contains Plica circularis covered by villi of varying heights. Crypts of lubricant are present which contain absorptive columnar cells. An important point that in submucosa there are no Brunner glands over here. So this is the section. You can see enteroendocrine cells, the enterocytes over here, the goblet cells, and the Pennant cells over here. Goblet cells first appear in duodenum. Their number increases in jejunum. Secretory granules of Pennant cells contain lysosomal enzymes, maintain normal bacterial flora of the small intestine. And the enteroendocrine cells secrete hormones that control various activities of the intestinal tract. So jejunum, in short, submucosa, we consist of the moderately dense connective tissue with blood vessels and mesenteric plexus and lymphatics. No Brunner gland. Important point over here, important negative finding over here. Then muscularis externa and serosa. The diagrammatic presentation again, this is the Plica circularis, this is the fold of the mucosa with core of the submucosa, the villi, the intestinal gland, this muscularis mucosae, the lamina propria, the submucosa and muscularis externa, and then the serosa. So same diagram showing all the same features as I have already described you. Next is illeum. Illeum contain absorptive columnar epithelium as in other parts of small intestine, contain few villi which are short and finger-like. Lymphatic lamina propria contains the lymphatic aggregation called the Peyer patches. Especially these Peyer patches are present on antimesenteric border, and these are large aggregates of the lymphatic nodules which are present in the lamina propria, and this is your identification point of the illeum. So these are the villi, and this is the area here I have already described that the M cells are present. So the epithelium which covers the area above the Peyer patch, this is the microfold, and the epithelial cells over here called M cells are microfold cells which are modified enterocytes, and these cells acting as antigen presenting cells capture any antigen present in the lumen of the intestine and present it to the lymphocyte in the underlying lymphatic nodule. So this is again very beautiful diagram. These are the villi covered by the simple columnar epithelial cells with the goblet cells. You can also notice that as we go down the intestine, the number of the goblet cells starts increasing over here. These are intestinal glands or crypts of lubricant. This is the muscularis mucosae, and this is the lymphatic nodule. Over here M fold is present. Again, another important diagram showing the lymphatic nodule or the Peyer patches in the lamina propria. So these are the references. Here I complete our today's class, and references are the Basic Histology by Laikus and Sadi, Histology text and atlas by John Quera, and Histology text and atlas by H. Ross. Thank you all dear students for your attention over here for a prolonged lecture today, and we will meet in the next class regarding the histology of large intestine. Till then, Allah Hafiz.